morning to one and all present here. Today class I am going to talk about the process of DNA packaging. Okay, so in this first I am going to explain about the general features of DNA. Okay, so what is mean by DNA? What is the full form of DNA? Yes, so DNA is one type of nucleic acid. Yes, no? So there are two different types of nucleic acids are present. One is RNA, another one is DNA. So today class I am going to talk about only the DNA general characters as well as and how the DNA is present inside the nucleus. Okay, so that things only today class we are going to understand. Okay, so deoxyribonucleic acid, which is the essential and which is the most important chemical compound, okay, which is carries the genetic information from generation to generation. Okay, so we are all know about the DNA is the genetic material which is present inside the nucleus. That is one important thing here. Okay, understand? So DNA is a genetic material which is present inside the nucleus. Okay, so who first proposed the DNA model? Any idea? Watson and Crick. Then who first give this DNA structure? Mixture. So the mixture is first discover the structure of the DNA. Okay, so who first identified the base pairs of DNA? Who first identified the base pairs of DNA? Tell me the answer. So the structure of the DNA first time discovered by the mixture. Okay, in the year 1869. Okay, so the base pairs of the DNA. What are the four nitrogen bases are present in DNA? Means one is adenine, cytosine, guanine, and thymine. Okay, this was first proposed by Albert Koshel. Okay, in the year 1881. Okay, in the year 1881, Albert Koshel first discovered the four base pairs of the both DNA as well as RNA. So both are, we call it as a nucleic acid, no? So the both nucleic acid nitrogen bases are discovered by Albert Koshel. So the what are the four nitrogen bases are present in the DNA structure? Means one is adenine, cytosine, guanine, and thymine. Okay, understand? In RNA, which is replaced by uracil, that is the thymine is replaced by the uracil. Okay, and the next is Erwin Chargaff. Okay, Erwin Chargaff. This person, it formulate the two rules. Okay, it formulate the two rules. One is the amount of adenine equal to the amount of thymine. Okay, understand? The always the adenine is paired with thymine. Cytosine is always paired with guanine, no? So the amount of adenine equals to the amount of thymine, but the amount of guanine equals to the amount of cytosine. Okay, understand? The amount of adenine equals to the amount of thymine. The amount of guanine equals to the amount of cytosine. Okay, and the amount of purines. Okay, the amount of purines is equal to the amount of pyrimidines. Okay, so this is the first rule of the Chargaff. Okay, this is the first rule of the Chargaff. Okay, so before that we know about the chemical structure of the DNA. So what are the chemical components are present in the DNA? Means, so there are three components are present in the DNA. One is phosphoric acid, sugar, and nitrogen bases. Okay, so now sugar is known as deoxyribose sugar. Okay, and another one is phosphoric acid, and third one is nitrogen bases. There are two different kinds of nitrogen bases are present. One is pyrimidine, another one is purines. Okay, the examples of pyrimidines. What are the examples of pyrimidines? Thiamine and cytosine. Thiamine and cytosines are the examples of pyrimidines. What are the examples of purines? Adenine and guanine. Adenine and guanines are the examples of purines. Okay, understand? So the amount of purines are equal to the amount of pyrimidines. Okay, so this is the first rule of the Erwin Chargaff. Okay, the second rule of the Erwin Chargaff is the amount of adenine and thiamine does not equal to guanine and cytosine. Only the amount of adenine equals to the amount of thiamine. Okay, but the amount of adenine and thiamine does not equal to guanine and cytosine. Okay, so these two rules are formulated by Chargaff. Okay, understand? And this person is the Erwin Chargaff. Okay, so next in this slide we are know about the Rosalind Franklin, the contribution of the Rosalind Franklin. Okay, this Rosalind Franklin used X-rays crystallography techniques to find out the 3D shape of the molecules. 3D shape of the molecules. Okay, this is the one important work by this Rosalind Franklin. Okay, in this work only she got the structure of the DNA. Okay, she applied this technique to different samples including DNA and observed X-shaped structure of DNA. X-shaped structure of DNA. Okay, so the Rosalind Franklin first time identified the X-shaped structure of the DNA. Okay, by using the technique crystallography. Okay, and next we are all know about the two scientists named Watson and Crick. Okay, so these two persons are based on the Rosalind Franklin and Wilkins work. Based on the Rosalind Franklin and Wilkins work, these two persons proposed the double helix model of DNA. Okay, according to them, this double helix model of DNA, it's contain two polynucleotide chain, the strands, two polynucleotide strands which are run anti-parallel to each other. Anti-parallel to each other. That means one strand will be 3' to 5' another strand will be 5' to 3' Okay, so this is the primers strands of the DNA. Okay, so the Watson and Crick, it's given the structure of the double helix model of DNA. So according to them, the DNA it's contain two polynucleotide. Okay, so why it is called it as a polynucleotide means there are four different nitrogenase bases are present in their structure, that is adenine, thiamine, cytosine and guanine. Okay, so this that's why that DNA is called it as a polynucleotide. Why the DNA is called it as a polynucleotide means there are four different types of nucleotides are present: adenine, thiamine, cytosine and guanine. Okay, understand this nucleotides it's run always in an anti-parallel manner. Okay, so each strands are run anti-parallel. That one will be 3' to 5' end, another will be 5' to 3' end. Okay, so this was proposed by Watson and Crick. For this work, the two persons are got a Nobel Prize. Okay. Okay, understand? So this is about the history of the DNA. So next we are going to see about the different types of DNA. So there are three different types of DNAs are present. One is A DNA, another one is B DNA, and third one is Z DNA. So there are three different types of DNAs are present. Okay, so next we are going to understand about the chemistry of DNA. So there are three different types of chemical molecules are present in the DNA. One is pentose sugar. One is pentose sugar, which is known as deoxyribose. The another one is a phosphoric acid. Okay, so DNA is a negatively charged or positively charged molecule. DNA is a negatively charged or positively charged? Yes, very good. DNA is a negatively charged because of the presence of phosphate group. Okay, in DNA it's having a phosphate group. Okay, the phosphate groups are the backbone of the DNA. Okay, phosphate groups are the backbone of the DNA. Okay, understand? So that's why the DNA is a negatively charged. DNA is a negatively charged. Okay, and also one sugar group is present. The name of the sugar is deoxyribose. Okay, and third important chemical compound is a nitrogen bases. So there are four different types of nitrogen bases are present: adenine, thymine, cytosine, guanine. Okay, so which are studied under the two terms. One is pyrimidine, another one is purine. Okay, so that's why DNA is called it as a polynucleotide. Polynucleotide. Okay, so this is about the chemistry of the DNA. Okay, understand? Any doubt in this topics? No. So these are the structures of the DNA. Okay, so in this we know about the two important topics. One is nucleoside, another one is nucleotide. Okay, so we want to here know about the difference between nucleoside and nucleotide. Okay, so what is the meaning of nucleoside means? So in nucleoside only pentose sugar and nitrogenase bases are present. Okay, so in DNA structure only pentose sugar and nitrogenase bases only present means that structure is called it as a nucleoside. Understand what is the meaning of nucleoside? Nucleoside means pentose sugar and nitrogenase base. So here it is a DNA, no? So which kind of sugar is present means deoxyribose. So deoxyribose plus adenine. Okay, so this structure is called it as a nucleoside. And what about nucleotide? Pentose sugar, nitrogenase base and phosphate group. Yes, very good. Phosphate group. These three components are present. Structure is called it as a nucleotide. Okay, if nucleotide means there are three different chemical compounds are present. One is phosphate group, another one is deoxyribose sugar, and third one is nitrogen base. Okay, so these three structures are present means which is called it as a nucleotide. Understand? So this is the general features of the DNA. Okay, so next we are going to see about the process of DNA packaging. Okay, process of DNA packaging. Okay, so why DNA is packaging? Why the DNA is present in a packaging manner inside the nucleus? Yes, very good. What is the length of the DNA in a haploid genome? In a haploid genome, the length of the DNA is around 3 meters. Okay, around 3 meters. Understand? The length of the DNA in haploid genome is around 3 meter. What is the size of the cell? The size of the cell is microscopic nature. Even the nucleus is very smaller. It's compared to that cell. Okay, the nucleus size is, it is a micrometer in diameter. Okay, so in order to fit the DNA inside the nucleus, it create a what? It create a more compressed and compact structure. More compressed and compact structure. Okay, so that's why the DNA is present in a packaging manner inside the nucleus. Understand? So in a haploid genome, the length of the DNA is 3 meter. Okay, so in order to fit the nucleus, okay, in order to fit the nucleus, the DNA is to be make the process of DNA packaging to create the more compact and condensation structure, that is call it as a chromatin. That is call it as a chromatin. அதாவது DNA ஓட சைஸ் பாத்தீங்கன்னா உங்களுக்கு 3 மீட்டர்ஸ் இருக்கு. நியூக்ளியஸ் ஓட சைஸ் பாத்தீங்கன்னா உங்களுக்கு மைக்ரோமீட்டர் இன் டயாமீட்டர்ல இருக்கறனால அது வந்து ஃபிட் ஆகணும் அப்படிங்கிற ரீசன்காக என்ன இருக்குன்னு பாத்தீங்கன்னா DNA வந்து chromatin அப்படிங்கிற ஸ்ட்ரக்சரா அது மாதிரி DNA குள்ள பிரசன்ட் ஆயிருக்கு. Okay, so this is about the DNA packaging. Okay, so this DNA packaging is present in 3 orders. Okay, 3 ஆர்டர்ல வந்து அந்த DNA packaging வந்து நடக்கும். So the first order of DNA packaging is nucleosome. The first order of the DNA is a nucleosome. The second order of the DNA packaging is solenoid fiber. The second order of the DNA packaging is solenoid fiber. And third order of the DNA packaging is Sculford loop chromatids. Okay, that is chromatids are present in a loop-like structure. Okay, in that first order, DNA packaging is a nucleosome. The second order of the DNA is a solenoid form, and third order of the DNA packaging is a loop-like structure. Okay, loop-like structure. So these are the 3 orders are present in the DNA packaging. Okay, so how the processing of DNA is takes place means generally the cells are produce a histone protein. Generally வந்து ஒரு செல் பாத்தீங்கன்னா it produce a histone protein. So histone proteins are positively charged. Histone proteins are positively charged. The DNAs are negatively charged. Okay, you generally cells are produce a histone protein. It is a positively charged. Okay, and DNA is a negatively charged. Okay, so the histone protein is play a important role in the DNA packaging. So the histone protein is very important for DNA packaging. So first we are know about the what are the histone proteins are involved in the DNA packaging. Okay, so in histone proteins are responsible for DNA packaging. Okay, so the DNA wrappers around the histone. Why it is wrappers mean it is attraction is takes place between two because the histone is a positively charged protein, DNA is a negatively charged protein, so automatically and positive and negatives are attracted. Yes, no? So the histone the DNA wrappers around the histones. Okay, so histones are organized to form a unit of 8 molecules called histone octamer. Okay, supposed to be it is a histone protein. So there are 4 histone proteins are present. One is H2A, H2B, H3 and H4. So these are the 4 histone proteins involved in the DNA packaging. These 4 histone proteins are it's make a octamer structure. Octamer structure. Okay, for example, H2A protein. Suppose is to be this is ball like structure, no? So one ball we considered as a H2A protein. Okay, in H2A protein, behind that one more H2A protein is present. Okay, likewise in H3B histone protein, one more H3B protein is behind that. Okay, and H3 histone protein, one more H3 protein is behind that, and finally H4 protein, that H4 protein is behind the one more H4 proteins. Okay, so the two copies of H2A protein and H3B, H3 and H4 proteins are present. So it form a 8 octamer histone protein. Okay, so this 8 octamer histone protein, it's involved in the DNA packaging. Okay, so histones are a set of positively charged basic proteins. Okay, so here histones are some acidic nature of histone proteins are present, but basic nature of histone proteins only involved in the process of DNA packaging. Okay, so histone proteins are set of positively charged basic proteins. Okay, so histones are rich in amino acids, lysines and arginine. Understand? So histones are rich in amino acids, lysines and arginine. Okay, so these three type of structures are present in the histone. Okay, so both the amino acids carry positive charge in the side chains. Okay, so in histone protein structure, amino acids, lysines and arginines are present, no? But particularly this amino acids are, it carries the positively charges in the side chains. Okay, this positively charged allow them to closely associate with negatively charged DNA through electrostatic interaction. So through this electrostatic interactions, the positively charged amino acids are allow them to closely associate with negatively charged DNA. So that's why the DNA wraps around the histones. Understand? Any doubt here? No. Next, neutralizing the charges in the DNA allows it to be became more tightly packed. Okay, so usually and the packaging எப்படி இருக்கும்னா அந்த ஆக்டோமரை சுத்தி இருக்கிற அந்த DNA நல்லா டைட்டா ராப்பரிங் ஆயிருக்கும். ஓகேவா, நல்லா டைட்டா வந்து ராப்பரிங் ஆயிருக்கணும். ஸோ இங்க வந்து two kinds of structures we can identify. One is loosely packed one and tightly packed one. Loosely packed one and tightly packed one. Okay, in acetylation and phosphorylation. Acetylation and phosphorylation. These two process are, it's affect the DNA packaging. Okay, these two the two process. One is acetylation, another one is acetylation, another one is phosphorylation. It affects the DNA packaging. Okay, and acetylation and phosphorylation, it makes the DNA more negatively charged. Okay, so that's why it form the DNA is loosen the packaging of DNA. So the DNA என்ன ஆகும்னு பாத்தீங்கன்னா அந்த packaging வந்து லூஸ் ஆயிடும். எதனாலனா phosphorylation and acetylation இந்த ரெண்டு process நடக்கறனால. So acetylation and phosphorylation, these two process, it affects the DNA packaging. Okay, so என்ன பண்ணும்னா DNA வை வந்து more negative ஆக்கிரும். DNA வை usually DNA is a negatively charged one. Okay, generally, but these two process are it more negatively charged DNA. So என்ன ஆகும்னா அந்த packaging வந்து loose ஆகிறதுக்கு நிறைய chances இருக்கு. So one enzyme acetyl transferase add the acetyl groups to the histone. So அந்த acetyl transferaseங்கிற enzyme acetyl group வந்து histone-ஓட bind பண்ணும்போது acetylization நடக்கும். acetylization and phosphorylation process வந்து occur. Okay, so இது என்ன ஆகும்னு பாத்தீங்கன்னா உங்களுக்கு அந்த DNA packaging tight ஆகறத அப்படியே இது லூசனிங் பண்ணிரும். ஓகே, யூசுவலா அது லூஸா இருக்க கூடாது, நல்ல டைட் பேக்கேஜிங்கா இருக்கும். ஸோ டிஎன்ஏவை மோர் நெகட்டிவ் ஆகும்போது ஆட்டோமேட்டிக்கா அது அந்த டைட்டனிங் வந்து ரொம்ப லூஸ் ஆகும். ஓகே, ஸோ திஸ் இஸ் அபவுட் தி ஹிஸ்டோன் protein. Okay, so there are 4 different types of histone proteins are involved in the DNA packaging. One is H2A, H3B, H3 and H4. Okay, these 4 proteins are present in two copies. So that structure is called as a octomer. That structure is called as a octomer. Histone proteins are positively charged, DNAs are negatively charged, so the DNA wraps around the histone. Okay, so the phosphorylation and acetylization, these two processes are affects the DNA packaging. Okay, so what is the main function of histone protein means the histone proteins are basic proteins that associate with DNA in the nucleus and helps to condense into a chromatin. Okay, so in the DNA it fit in the nucleus, no, so it is present in a chromatin fiber like structure. Okay, this chromatin fiber like structure is made with help of this histone protein. So the another function of histone protein is it takes place in the chromosome formation. Okay, the histone proteins, it take part in the chromosome formation. And another important function of histone protein is histone, it helps the DNA to fit inside the nucleus. Okay, and some of the histone proteins are act as a fool for the thread like DNA to wrap around. அதாவது பூல்ங்கிறது என்னன்னு பாத்தீங்கன்னா மேல ஒரு சென்டர்ல வந்து ஒரு ஸ்டாண்ட் இருக்கும். ஸோ அந்த ஸ்டாண்டுக்கு மேல ஒரு ரவுண்ட் ஷேப்ல வந்து ஒரு ஸ்ட்ரக்சர் இருக்கும், கீழே ரவுண்ட் ஷேப்ல ஒரு ஸ்ட்ரக்சர் வந்து பிரசன்ட் ஆயிருக்கும். ஓகேவா, ஸோ இது எப்படி இருக்குன்னு பாத்தீங்கன்னா நம்ம இந்த தறி இந்த பவர் லூம்ப் இருக்குல்ல, ஸோ அதுல பாத்தீங்கன்னா அந்த நூலெல்லாம் சுத்தி வச்சிருப்பாங்க இல்லையா, ஸோ அதை தான் நம்ம பூல்னு சொல்லுவோம். ஸோ மேல ஒரு சர்க்கிள் மாதிரியான ஒரு ஸ்ட்ரக்சர் இருக்கும், கீழே ஒரு சர்க்கிள் மாதிரியான ஸ்ட்ரக்சர் இருக்கும், சென்டர்ல ஒரு ஸ்டாண்ட் இருக்கும். அந்த ஸ்டாண்ட் சுத்தி என்ன பிரசன்ட் ஆயிருக்குன்னா த்ரெட் சுத்தி வச்சிருப்பாங்க, அதுதான் பூல்னு சொல்லுவோம். ஸோ அந்த மாதிரி ஒரு டைட் பேக்கேஜிங்கா டிஎன்ஏ பூல் மாதிரியான ஒரு ஸ்ட்ரக்சர் பிரசன்ட் ஆகிறதுக்கு இந்த histone proteins are help. Okay, so these are the main functions of the histone protein. Okay, so one, it take part in the chromosome formation and it helps the DNA to fit inside the nucleus. And some of the histone proteins are function as a pull for the thread-like DNA to wrap around. And the main function is the histone proteins are the basic that associate with DNA in the nucleus and help to condense into chromatin. Okay. So in this topic we wants to know about the term nucleosome. Okay, nucleosome. So nucleosome means the octomer structure of the histone protein. Octomer structure of the histone protein is called it as a nucleosome. Okay, so octomer structure means there are 4 proteins are involved, no, H2B, H3B, H2 and H4. So these 4 proteins are present in a octomer like structure. Okay, so this is the octomer H4 protein is present. Okay, so this structure is called it as a octomer. This structure is called it as a octomer. So that octomer structure is called it as a term of nucleosome. Okay, so the typical nucleosome, it's contain 200 base pairs of DNA helix. So in DNA helix, okay, 200 base pairs of nucleosomes are present. Okay, in histone octomers are closely contact and DNA is coiled on the outside of the nucleosome. So nucleosome outside la da DNA eppadi irukku pathina nalla tight ah wrapper ஆகி present ஆயிருக்கும். Okay, so nucleosome is nothing but octomer structure of the histone protein. So on the octomer structure of the histone protein ku outside la DNA வந்து நல்லா tight ah wrapping ah present ஆயிருக்கும். Okay, so the DNA makes two complete parts around the histone octomer. Okay, and the histone octomer and the DNA வந்து two turns la வந்து wrapper பண்ணிருக்கு. ஓகேவா? சோ இந்த மாதிரி two turns ல வந்து என்ன இருக்குன்னு பாத்தீங்கன்னா அந்த DNA வந்து wrapper ஆகி present ஆயிருக்கு. ஓகே, சோ இந்த DNA ரொம்ப டைட்டா தான் பேக் ஆயிருக்கணும் இல்லையா? சோ இப்போ என்ன ஆகும்னா the two turns are shielded off by an H1 molecules. H1 molecules. So அந்த ரெண்டு முடிச்சிருக்கு இல்லையா? இந்த ரெண்டு முடிச்சு கலண்டாம இருக்கிறதுக்கு one more histone protein is helped. Okay, the name of the histone protein is H1 molecules. H1 molecules. Okay, so this H1 molecule is not inherited. Not inherited. Okay, so that's why this histone protein is not including the octomer like structure. ஏன் அந்த octomer like structure ல நம்ம இந்த H1 protein நாம என்ன பண்ண மாட்டோம்னு பாத்தீங்கன்னா include பண்ண மாட்டோம். ஏன்னா இது வந்து non inherited. Okay, இது H1 proteinங்கிறது non inherited. மீதி இருக்கிற octomer structure ல இருக்கிற நாலு protein வந்து inherited. ஏன்னா DNA கூட wrapping ஆ இருக்கறனால. சரியா? சோ அந்த ரெண்டு டர்னோட சீலும் ஓபன் ஆகாம இருக்கிறதுக்காக இந்த H1 protein வந்து இன்வால்வ் ஆகும். ஓகே, சோ ஃபர்ஸ்ட் ஆக்டோமர் லைக் ஸ்ட்ரக்சர்ல 4 ஹிஸ்டோன் புரோட்டீன் இருக்கும். அந்த wrapper ஆன DNA ரெண்டு சீலும் வந்து ஓபன் ஆகாம இருக்கிறதுக்கு H1 proteins வந்து ஹெல்ப் பண்ணும். ஓகே, சோ திஸ் இஸ் about the nucleosome. Okay, so nucleosome is nothing but octomer structure of the histone protein. Okay, so in here additionally H1 protein is involved. So what is the main function of this H1 protein means the two turns are shielded off by an histone molecules. Okay, so next we want to know about the linker DNA here. Okay, so here the octomer like structure is constituently it is developed. அப்படியே அடுத்தடுத்து தொடர்ந்து அந்த octomer like structure வந்து ஃபார்ம் ஆயிட்டே தான் இருக்கும். அது வந்து கான்ஸ்டிடியூஷனா அது அப்படியே ஃபார்ம் அப்படியே கண்டினியூவா next that octomer like structure is formed. Okay, so here see here this is one octomer like structure. So this is one octomer like structure. This is one octomer like structure. Okay, so between the two octomer or between the two nucleosome, additionally 38 to 53 base pair long DNA strand is present. Okay, that is that what we call it as a linker DNA. Understand? So between the two nucleosomes, 38 to 53 base pair long DNA strand is present. Okay, that is called it as a linker DNA. அததான் நம்ம linker DNA ன்னு சொல்றோம். linker DNA ங்கிறது ஒரு நியூக்ளியோசோமையும் இன்னொரு நியூக்ளியோசோமையும் கனெக்ட் பண்ற DNA is called it as a linker DNA. So in that linker DNA how many base pairs are present means 38 to 53 base pairs. In nucleosome 200 base pairs are present. Okay, in linker DNA 38 to 53 base pairs are present. Okay va, ஒரு நியூக்ளியோசோம்ல பாத்தீங்கன்னா உங்களுக்கு அந்த நியூக்ளியோசோம்ல இருக்கிற டிஎன்ஏல பாத்தீங்கன்னா 200 base pairs வந்து present ஆயிருக்கும். But in the case of linker DNA 38 to 53 base pairs வந்து present ஆயிருக்கும். Okay, understand? Which is associated with that linker DNA is associated with H1 histone protein. Okay, H1 histone protein. So always the linker DNA is associated with the H1 protein. Okay, so this is, we call it as a linker DNA, no? So this octamer like structure is called it as a core DNA. Core DNA, understand? The linker DNA, it's always present between the two nucleosome. The individual one nucleosome is called it as a core DNA. Core DNA, understand? ஒரே ஒரு நியூக்ளியோசோமை மட்டும் நம்ம என்னன்னு சொல்றோம்னா கோர் டிஎன்ஏ. ரெண்டு நியூக்ளியோசோம்க்கு இடையில இருக்கிற டிஎன்ஏ வந்து நம்ம லிங்கர் டிஎன்ஏ. ஓகே, so this is about the linker DNA and nucleosome. So next we are going to know about the chromatin fiber. So the DNA it's present in a chromatin like structure, no? So inside the nucleus. So inside the nucleus the DNA is present in a chromatin fiber. Okay, so the nucleosomes constitute the repeating unit of a structure. Okay, so nucleosome repeating என்ன பண்ணும், கான்ஸ்டிடியூட் பண்ணிட்டே இருக்கும், ரிப்பீட்டிங்கா அந்த ஸ்ட்ரக்சரை வந்து ப்ரொடியூஸ் பண்ணிட்டே இருக்கும் இன்சைட் த நியூக்ளியஸ். ஸோ இன்சைட் தட் நியூக்ளியஸ், தட் நியூக்ளியோசோம் கான்ஸ்டிடியூட் தி repeating unit of a structure. Okay, we see when we viewed under the electron microscope that repeating nucleosome is appeared as a beads on string. Okay, ஒரு நூல்ல மணி கோக்குறோம் இல்லையா, அந்த மணி கோத்த மாதிரியான அந்த நியூக்ளியோசோம் ரிப்பீட்டா ஒரு ஸ்ட்ரக்சரை ஃபார்ம் பண்ணிட்டே வந்துச்சு இல்லையா. ஸோ இந்த மாதிரி ரிப்பீட்டடா என்ன பண்ணிட்டே வரும்னா நியூக்ளியோசோம் வந்து ஃபார்ம் பண்ணிட்டே இருக்கும் நியூக்ளியஸ்குள்ள. ஸோ அது வந்து எலக்ட்ரான் மைக்ரோஸ்கோப்ல நம்ம வியூ பண்ணும்போது, ஓகே, when we viewed under the electron microscope the repeating unit of nucleosome it appears like a beads on string. பார்க்கிறதுக்கு அப்படியே நூல்ல மணி கோத்த மாதிரியான ஒரு ஸ்ட்ரக்சரை நம்ம வியூ பண்ணலாம். ஓகேவா? This structure we can see in the leptotin stage. Okay, leptotin stage. So leptotin stage is a one space of the phase 1 of the meiosis 1. So meiosis it has two phases. One is meiosis 1 and another one is a meiosis 2. So in meiosis 1 the first stage is prophase 1. In prophase 1 leptotin stage is a first stage. Okay, in this leptotin stage the chromosomes are appear as a beads on string. Why it is appear like this means the nucleosome constitute the repeating unit. அது வந்து ரிப்பீட்டடா அந்த மாதிரியான ஸ்ட்ரக்சர் அந்த நியூக்ளியோசோம் ஃபார்ம் பண்றதுனால தான் நமக்கு leptotin stage prophase 1ல மியாசிஸ்ல prophase 1ல leptotin stageல நம்ம beads on string அப்படிங்கற ஒரு ஸ்ட்ரக்சரை நம்ம மைக்ரோஸ்கோப்ல பார்க்கும்போது வியூ பண்ணலாம். ஓகே, அண்டர்ஸ்டாண்ட்? ஸோ நியூக்ளியோசோம் ரிப்பீட்டடா இது பண்ணி beads on string மாதிரியான ஒரு ஸ்ட்ரக்சர் தெரியும். ஓகே, நெக்ஸ்ட் H1 of one nucleosome interact with the H1 of neighboring nucleosome. Okay, so always அந்த H1ங்கிறது எதுக்கு ஹெல்ப் பண்ணும், அந்த DNA ரெண்டு டர்னா ராப் பண்ணியிருக்கும். ஸோ அந்த ரெண்டு டர்ன் வந்து சீல் வந்து ஓப்பன் ஆகாம இருக்கிறதுக்கு அது நல்லா டைட்டா சீல் பண்றதுக்கு H1 protein ஹெல்ப் பண்ணும் இல்லையா. ஸோ ஆல்வேஸ் the H1 of one nucleosome of the fiber. So in the H1 protein neighboring H1 proteins அப்படியே என்ன பண்ணுனா long chain அது அப்படியே interact பண்ணிட்டே இருக்கும், associate பண்ணிட்டே இருக்கும். So அப்ப என்ன பண்ணுனா ஒரு fiber like folding like structure வந்து form பண்ணும். Okay, so the size of the fiber is 30 nanometer. Okay, the size of the fiber is 30 nanometer. Okay, so this structure is called as a solenoid structure. Okay, this is the second order of the DNA packaging. So the first order of the DNA packaging is nucleosome. The second order of the DNA packaging is a solenoid structure. Okay, so solenoid structure means the 30 nanometer fiber like chromatin is formed. No, that structure is said to be solenoid structure. Okay, this solenoid structure is having 6 nucleosome per turn. Okay, so அது ரெண்டு turn ஆகுது. DNA வந்து ரெண்டு turn ஆ wrapper பண்ணிருக்கு இல்லையா, அதுல ஒரு turnல okay in one turn 6 solenoid structures வந்து present ஆயிருக்கு. ஒரு turnக்கு 6 solenoid structures are present. Okay, so DNA is a solenoid and packed about 40 folds. ஒரு DNA ல பாத்தீங்கன்னா உங்களுக்கு 40 folds of DNA solenoid structures வந்து present ஆயிருக்கு. Okay, so DNA is a solenoid and packed about 40 folds. 40 folding அந்த DNA வந்து அந்த solenoid structure குள்ள present ஆயிருக்கு. Okay, so this is about the chromatin fibers. Understand? Any doubt? So chromatin fiber is nothing but the nucleosome. It constitutes the repeating unit of a structure. Okay, so when we viewed under electron microscope, the repeating unit structure is appeared as beads on string. Okay, beads on string. Okay, so this in the beads on string மாதிரி இருக்கிற ஸ்ட்ரக்சரை தான் நம்ம என்னன்னு சொல்றோம்னு பாத்தீங்கன்னா அந்த solenoid structure னு சொல்றோம். Okay, so the H1 of one nucleosome interact with the H1 of the neighboring nucleosome, resulting in the further folding of the fiber. So fiber and the chromatin fiber form பண்றதுக்கு இந்த H1 protein வந்து help பண்ணும். So 30 nanometer fiber வந்து அந்த ஒரு folding ல இருந்து develop ஆகும். So அத நம்ம solenoid structure சொல்றோம். So each that solenoid structure is having a 6 nucleosome per turn. Okay, ஒரு turnல 6 nucleosomes வந்து அந்த solenoid structureல present ஆயிருக்கு. So அப்ப solenoid structureங்கிறது nucleosome, core DNA, linker DNA வும் present ஆயிருக்கு. Core DNA and linker DNA. Okay, so DNA is a solenoid and packed about 40 folds. Okay, this structure is stabilized by interactions between the different H1 molecules. Okay, and the structure அப்படியே stabilized இருக்கணும். unstabilized ஆயிருச்சுனா tightening வந்து போயிடும். So stabilized இருக்கணும். அந்த stabilized இருக்கிறதுக்கு H1 molecules will be helped. Okay, understand? So in this way the processing of DNA is takes place particularly in eukaryotic cell. So particularly ஒரு eukaryotic cell ல பாத்தீங்கன்னா so இந்த மாதிரி தான் அந்த DNA packaging வந்து develop. Okay, so additionally in this class we want to know about the he wants to know about the non-histone chromosomal proteins. Okay, some of the basic nature of the histone proteins are involved in the DNA packaging. Okay, the acidic nature of the histone proteins also made by the cells. That acidic nature of the histone proteins are not involved in the DNA packaging. So that kind of histone proteins are called it as a non-histone chromosomal proteins. Non-histone chromosomal proteins. These are all acidic in nature. Okay, the basic nature of histone proteins only involved in the DNA packaging. The non-histone proteins did not involve in the DNA packaging. Okay, it is acidic in nature. euchromatin and heterochromatin. Euchromatin and heterochromatin. Woodard is one scientist. Okay, Woodard is one scientist. That person was find out the thicker and dark region of the nucleosome. Okay, dark region of the nucleosome. So nucleosome means it is a octamer structure of the histone protein. Okay, so in this octamer structure of the histone protein, the Woodard, it's find out the darker portion and tightly packed regions. Okay, that regions we call it as a, and the region we call it as a euchromatin. Okay, euchromatin, and the chromatin that is tightly packed, it is called it as a heterochromatin. So the loosely packed chromatin is called it as a euchromatin. The tightly packed chromatins are called it as a heterochromatin. Okay, so in this usually the euchromatin region, usually euchromatin region protein synthesis takes place. Okay, in this euchromatin region the protein synthesis takes place. Okay, so during that protein synthesis the DNA is makes to mRNA. DNA is mixed to mRNA. Okay, so this particular process takes place in the euchromatin region of the this chromatin fiber. So in the chromatin fiber la euchromatin region la da pathina ungalku protein synthesis nadakkum. So protein synthesis அப்ப என்ன நடக்கும்? first step என்னன்னு பாத்தீங்கன்னா DNA, the double stranded DNA is converted into single stranded mRNA. Okay, this process takes place in the euchromatin region. Okay, so heterochromatin is a tightly packed region, euchromatin is a loosely packed region. So on the loose packing ஆயிடுச்சுனா தான் DNA வந்து unbinding ஆகும். Okay, unbinding ஆகி it became a mRNA. Okay, so that's all about the process of DNA. Any doubt? No doubt. So how many base pairs are present in that nucleosome? 200 base pairs. So Tamal Selvi, what is meant by linker DNA? Okay, Satish, what is meant by core DNA? What is meant by core DNA? சத்தமா சொல்லு. the nucleosome DNA wrapper பண்ணியிருக்கிற ஸ்ட்ரக்சரா core DNA. Okay. Why DNA needs the DNA and the packaging manner la present? Why DNA requires packaging? Why? Sona, சொல்லுங்க. So what are the histone proteins are involved in the DNA package? என்னென்ன histone proteins வந்து DNA package? அது nitrogen base pairs. நான் கேக்குறது histone proteins. 4 histone proteins வந்து DNA packaging ல இன்வால்வ் ஆகுதுன்னு சொல்றோம்ல. என்ன 4 histone